ahead and begin. So welcome to our webinar today. My name is Tom Rabe. I'm a research agronomist for General Mills. I support our oat supply chain and a lot of the activities associated with oats. Speaking of oats is a collaborative arrangement between the University of Minnesota sponsored Oat Global organization and also Charlene White from Agriculture and Agri-Food Canada's Oat newsletter organization. So we want to thank both of them for continuing the hard work to bring these monthly webinars out to our oat community. Thank you all for attending today. The title of our of today's webinar is Technological Advances that can accelerate oat improvement, and we have a great lineup of 3 speakers. First off will be Jorge Dubcovsky and Isabel Alicia del Blanco from the University of California, Davis, and they will start the presentation. Once they're completed, we are going to have a short time for a few questions, a little bit of Q&A, and then we'll move to the second speaker, who is Jaswinder Singh from McGill University in Canada. Also want to point out to please use the Q&A section that's located in Zoom or into the chat section as well. We'll be watching for those questions. And with that, I'm going to go ahead and turn that over to Jorge.
Okay, so let me share the screen. Let me know when you're seeing it.
I can see it. It's not in presentation mode. I don't know.
Yeah, I already put it in presentation mode, and let me change the display settings.
Yep, there we go. Well,
so presenter, I think that we are
clear. We're good.
Okay, thank you very much for the invitation to presenting this series of technological advances for oat improvement. But before I start, I would like to take 2 minutes just to introduce our oat breeding program, so people get familiar with the opportunities that we have over there to do collaborations in oat. So this program is mainly run by Alicia del Blanco. She has been the oat breeder for 9 years in the UC Davis. And California is the largest milk producing state in the US and grows a lot of forage, and among them 100,000 acres of oats to feed these animals. So oats are predominantly grown for forage, silage, hay, or green chop, but 10% of this acreage is also used as feed grain for livestock. But we're trying to expand a little bit the uses. So Alicia also has a white grain oat program, and for food products, we have been doing some collaborations to test for gluten epitopes and discard some materials that show some epitopes. So now we have a cleaner materials. We are also starting collaborations with a small company that's starting to produce oat milk in California. So we are looking for opportunities for collaborations. And with USDA, we are now starting from next year some disease nurseries in California, because we have very consistent yellow dwarf infections here, stem rust every year. And it's a good location to see entrance of new races from Central America in the United States. So for barley and other crops, we have nurseries that a lot of people in the US use to test their materials. We hope to do something similar with oats. So the breeding program is aimed at the release of commercial cultivars. So we have been releasing cultivars to California for the last 25 years. And we are focused on yield stability, short cycle to feed in rotations with other crops, disease resistance, mainly yellow dwarf virus and stem rust, high test weight and improve end-use quality, and enhance agronomic performance. And we hope to grow this program and to establish collaborations with others, maybe to generate national efforts, as we are doing in the wheat community, where we have the wheat collaborative agriculture projects, where we all come together to try to promote the technological developments in public breeding. So that's all for the program, and I will move to my presentation that is mainly on how to improve transformation technologies. So we were probably where the oat community is now. We have transformation, but the efficiencies were not that great in wheat. So we were looking for better technologies to improve regeneration. So we were working with a couple of transcription factors of 2 families. The first one is the growth regulating factor, or GRF family. These are transcription factors that are regulated by microRNA. So they have a binding site here where the microRNA degrade these in tissues where you don't need to produce cell division and growth. And this transcription factor works together with a cofactor that's called GIF. They bind together and they attract a chromatin remodeling complex. So basically the GIF is the docking port for the chromatin remodeling complex, and that promotes cell division, and a lot of cell cycle genes are regulated by these transcription factors. Because they are associated with growth in Arabidopsis, these genes have been shown to be very active in the shoot apexes. You can see here in situs, they are very abundant here, but also in the leaves. If you, this is an Arabidopsis leaf, you see cell division in this part of the leaf in Arabidopsis, and the GRF are expressed exactly in the same region and opposite to the microRNA 396. When you have microRNA 396, you don't have GRF. When you don't have microRNA, you have GRF. And if you make mutations in this part of the binding site, so the microRNA cannot degrade it anymore, then you have GRF in all the plant. And these genes are essential to maintain meristem identity, and that's why we thought they will be useful for transformation. So if you have mutations of the GIF1, you have smaller plants. The same thing if you overexpress the microRNA 396. But if you put the two together directly, you do not have a meristem at all. So these two genes are essential to maintain the homeostasis of meristems. And to be honest, we didn't start to work on these genes for regeneration efficiency. We started because it was reported in Arabidopsis that when you overexpress these genes, you can get larger organs, and we were trying to see if we can increase biomass in wheat. And if you combine the two, the GRF and the GIF, you get even more larger increases in organs. And they made a trick in Arabidopsis that was pretty interesting, that was to use a link in the two genes and express them together as a single protein. And this is even more efficient because the two factors find each other immediately, and they produce even larger organs in Arabidopsis. So we went into grasses. There are like 10 GRF genes in the wheat genome and 3 GIF genes. So we have to select which ones to work with. So we look at the expression. So this is the normal expression of genes like cyclin D that are involved in cell division. So this is the spike, the grain, the leaves, and the darker the blue is, the higher the expression. So these are the expression of the GIFs, and you can see very clear here that GIF1 is the one that has a pattern that is similar to the cyclin gene. So we selected GIF1. The GRF, there are many of them that have a good pattern, but we selected GRF4 because in rice, this gene has been shown to increase biomass. And here they started the story. So this is our normal transformation, in which you can see we get a 2%, 3% of the Kali are regenerating shoots. And to our surprise, when we did a transformation with this is agro transformation, almost every Kali was showing regeneration of shoots. It was completely amazing for us. We have never seen something like that. So we did now a lot of experiments. This is the average of 15 experiments. And we have above a 60% regeneration efficiency now in wheat compared to the 5%. And this is really a complete underestimate, because the way we are calculating the 60% frequency is the percent of Kali that produce at least one shoot. And as you can see here, each of these Kali produce several independent shoots. So if we would count that, probably we are in a 200% efficiency. But since we have not tested them to demonstrate that they are independent events, we are counting it in a very conservative way. So this really is kind of an underestimate. And you need to have the two genes. If you transform with GIF alone or with GRF alone, you don't get the efficiency of transformation that you get with the two of you. And the important part of this technology is in contrast with baby boomer and bushel, that when you use them, you get a lot of sterility, and then you need to work either with inducible promoters, or you need to select very careful promoters. So it took the maize industry a lot of years to find a way to go around the sterility produced by those genes. The GRF genes, because they are controlled by the microRNA 396, they are not expressed in the adult tissue, and the plants look normal, and they are fertile. So we don't have the initial problem that the baby boom bushel technology had. So we tested if it was important to have the two genes fused. So we created a construct that has the GRF and the chief each expressed by its own ubiquitin promoter, and compare it with the chimera. And the chimera has better efficiency than the other one. We also tested which GRF was the best one. So we fixed chief 1 and we test different GRF, and you can see here that GRF4 combination with chief 1 is the best one, 65% the regeneration efficiency. And in the chief, we did the same. We fixed GRF4, test the different chief, and chief 1 was the one that produced the largest efficiency. So the combination chief 1 GRF4 seems to be the best for wheat. And then we try to start using applications. So we combine this vector with the CRISPR-Cas9 technology. Sorry, my throat is a little bit dry. And we use a guide RNA for the gene Q. This is the gene in wheat that produces the free threshing of the grain, and also produce, when you mutate it, extra florets in the spikelets. So, and it's easy because there's no B genome copy. So if you knock out the copy in tetraploid wheat, you see the phenotype. And again, we get a high transformation efficiency. We get in a single experiment with 2 plates, 32 transgenic plants, and 96% of them were editing. That's because, well, maybe it's not that I'm not saying that the GRF technology increases the efficiency of CRISPR, but in this case we have a good guide RNA. What the GRF technology increases is the number of plants you recover there, and you can see, well, these are the type of mutations produced by CRISPR, and all the plants show the phenotype, so we are sure that we have produced the transgenics. And this has completely transformed the way we are doing research in wheat. So now we are CRISPRing dozens of genes. We have completed, we have probably, this was several months ago, so probably in the 800 independent events and 20 genes that we have knocked out. We can even now do promoter bashing, where you knock out multiple parts of the promoter to see the regulatory regions, because you get so many plants for just a couple of plates that now you can afford to do those kinds of experiments that were previously limited for Arabidopsis. So the other interesting aspect of this technology is that allows you to eliminate the antibiotic selection markers for those people that are concerned in transgenics that you have this type of selection markers. What we notice is you normally put your kali in auxin to induce the formation of kali. And if you want to get regeneration, you need to put cytokinin to produce the regeneration. But because these genes promote the synthesis of cytokinins, we see directly in the auxin media regeneration. So you can use this as a selection tool to find the transgenic plants. So we tested that. We did that one without any selection marker, and more than half of the plants were transgenic. So we have this method allows you to try to produce marker-free transgenic plants if that is your objective. So we tested in multiple genotypes in wheat. In wheat, we have very few genotypes that we can transform by normal bombardment technology, or even with agro. There's bob white, fielder, chronos in tetraploid. So we tested varieties that we were not able to transform before. And with this technology, we were able to transform it. We did the same in hexaploid wheat. These are 3 genotypes we have never transformed, and we were able to recover transgenic plants. Similar in triticales, we were able to transform it for the first time. You can see that we don't get that 60% efficiency we get in a few varieties. In others, we are, even with the GRF technology, we are getting 5%, 10%, but that is compared to 0%. And that is without playing at all with the media or doing any selection, just using the same media that we use for these varieties, just putting the construct with the GRF4 chief, we're now able to obtain plants for that. And the good news is we use that construct in rice, and we also showed a twofold increase in regeneration efficiency. So I'm pretty sure that this will work well in oats. We have not started yet. We were trying to get some genotypes, and we get some Baja genotypes from Poland now that we are growing to test. We have now several companies have licensed the technology to UC Davis, and we know that people in maize are already getting good results also with this technology. So in summary, this technology increases dramatically the frequency, the efficiency of transgenic. So if you don't have agroinfection, this will not help you. But if you have agroinfection, then the efficiency of regeneration is greatly increased. I would say in a conservative way, we increase it to 60% in the good varieties in wheat, and we were able to transform ones that we were not able. The plants are normal and fertile. You have a broader number of genotypes that is important when you want to apply CRISPR in breeding applications. The chimera, The chimera also produces shoots in the absence of cytokinins, so you can use plants without antibiotic selection. You can combine the TRF-GIF with CRISPR-Cas9. These vectors are available. We've deposited them in AdGene, so everybody can get them directly from AdGene. The chimera was useful in rice, as we have indications that it's useful in maize too, so you can use it probably in oats. Of course, you can select a TRF and a GIF directly from oat, and probably that will maybe more efficiently. But to start with, we will start with the wheat one. So we are growing the oat variety Bacha, that is the one that used in Poland for transformation, and successfully. So we wanted to start with a good genotype. And we have succeeded also in several dicot species, so we think that this could be a good solution also for oats. So that's it. These pictures are from Alicia. The oats are growing fast now. We harvest a lot earlier than the rest of the country, our oats. So, and the pictures are all from Alicia too. So I think they're pretty nice pictures. So I like to do my presentation short to have a time for questions. So let me know if you have any questions and I'll try to answer them.
I actually have a question. Thanks very much, Jorge. That was really great. And Alicia too, for all your work there. That's really exciting that this eliminates the antibiotic resistance marker use. And also,
of course, it's easier to do it with the antibiotic, but you can do it without the antibiotic, and you will get half of the plant's transgenics.
Right, and the idea that more of the genotypes now work. Do you have any idea of the mechanisms behind why some genotypes work and some just never do?
No, there are different things. One is infection with agro. So sometimes you don't get infection. When you don't get infection, this technology will not help you. And then it's difficult to make the tissue to start forming a meristem. So that's why the baby boom bushel have worked, because you prompt those cells to develop. So now we are doing some collaborations with the people in Corteva to see if we can combine the two technologies and how that will look like. You can also produce inducible promoters for this one if you want. You cannot. We have tested it for dicots, and I'm not sure we tested it in monocots. But you can, instead of using the GRF4, you can use a resistant form without the blocking the microRNA binding, and then the effect is a lot stronger. But then you produce abnormalities. But you can think of way of producing that with an inducible promoter. So this opens a new range of possibilities of combinations and technological things, because now we have one more tool.
Right, yeah, no, that's really exciting. And Jose Costa is asking, is this technology available to other researchers?
Yes, as I said, we've deposited all the clones are deposited in AdGene. So everybody can request it directly from AdGene and we'll get the construct. And if you, we have developed since then some for pepper and Solanaceae and tomato constructs, and for soybean and for maize constructs. Those are not deposited, but you can request it from us, and there's an MTA for testing. And even for companies, they can test it for free. But of course, if companies want to use it, they have a license, and some companies are already starting to take those licenses. They're not that expensive for a company. But for research, it's completely free and available, and we will be interested in helping or collaborating in anything that you want.
Oh, that's really great. Boy, you guys have been busy.
Yeah, I have a lot of support in my lab from HHMI, so we have resources to do the basic research. We are struggling always with the resources for the breeding part.
Right, yeah, I think is pretty typical.
Yeah.
We do have another question from Jaswinder Kaur. I was asking, you were regenerating transgenics without cytokinins?
As I said, yes, you start seeing regeneration. It's not as great as you will see it. You start in the initial formation of the shoots. What we do then is once we select those things, we put them with cytokinins to complete their regeneration. But you start seeing, let me see if I can, if I can show you again that slide. I don't know if you see it clearly. You don't get complete shoots there, but you get enough regeneration that you can excise those ones and then move it to another medium. The other thing that you can do is use very suboptimal concentrations of cytokinin, but again, you will have more escapes if you do it that way.
Cool.
Now we finally move it to cytokinins, but because we have regeneration initially without cytokinins, you can use it as a selection method.
Yeah, that is really interesting. We have another question here from Irfan Iqbal. Wonderful presentation. I have a question. If we order the constructs from AdGene, would they provide us the sequence of the constructs as well?
Yes, and if something is missing, you can always write us and we will try to help. Yes, they have the map of the vectors and all the things, and yeah, and also all the sequences. There's a pattern that we put there for licensing to companies, and all the sequences are there also too. But just send us an email and I'll send you whatever you don't find in the AdGene side. We will send it to you. We are committed to public use of the thing, so we will be glad that you use it.
Yeah, that's wonderful to hear. There aren't any other questions right now.
I have one quick question. I'm curious about the frequency of somal clonal variation.
Well, I have not measured it, but the good news is that instead of taking us 90 days to do the regeneration process, we have reduced it to 60 days. And as you know, the somal clonal variation is a result of the stress of the tissue culture. So we hope that this will reduce a little bit, but to be honest, we have not tested it. But again, you edit and then you backcross, so you always clean your backgrounds by backcrossing. So
right. Good. Should we move on to Jaswinder's presentation, and then I'm sure there may be more questions for Jorge and Alicia after his presentation.
So Jaswinder, are you ready to take over?
Yes, I can. Sure. Can I share the screen?
There we go. We can see it.
Sorry, I have to stop once and then I have to reshare it. So I'm sorry. There is something wrong here. Can you see it?
Yes.
Yes, I can see it.
Okay, fantastic. So should I start?
Please do.
Fantastic. So first of all, thank you very much, the Oat Global.
Oops, now we lost your audio.
Not good idea. Hello, can you hear me? Okay. So first of all, thank you very much, Oat Global. Actually, they are doing a very, very good job by organizing these seminars. I'm really, really happy to see all those. And actually, it's also improving my knowledge in oat as well. So I'm also thankful to Jorge for such a wonderful presentation and setting up a stage for me for the Oat transformation. Although it is always difficult speaking after Jorge, you know, where he is showing 60% or 80% type transformation. So we can't match that, but at least we already ordered his construct, so we have not used it yet. But I believe we will also improve our transformation using his technology in future. But what we are using at this stage, I will just present what we are doing to improve oats in my lab. So basically, my research focus in my lab is actually we are working on small grain cereals, and the idea here is to genetically dissect some of the important traits. And main work I have done mainly in barley, but also in wheat. Recently, my lab also moved towards oat, and thanks to Dr. Nick Tinker, who introduced me to the oat community. So basically, oat community is really a wonderful community and is very helpful community. And so with their help, currently in my lab, we are doing genetic transformation in oat, especially to understand the oat genome and also fine tune some of those important genes. So I don't think I need to explain oat that much. Many of the things are already mentioned by Jorge, and many of you also working in the oat side. But as you know, the oat is very, very important. It is used in multiple ways, grain forage, fodder. So it also have many, many good natural antioxidants, but mainly it is very, very now becoming a very important crop because of the soluble dietary fiber, and beta-glucan is playing an important role. And that makes oat a little bit more unique than other cereals. So in general, oat is also have proteins, oil, carbohydrates. But as I said, a more unique thing is that beta-glucan, which is present in high amount. And we now know that this beta-glucan actually reduce serum cholesterol and blood sugar. So basically, it is also considered, I believe in 2007, FDA, considered oat. Now they just put in the list that it's a health food. So, but in oat production or for the oat improvement, we have so many challenges, right? So we have challenges in the agronomy side, we have challenges in quality side to improve it further, and certainly there are many challenges in the disease side. So these are some of the challenges which we can, tackle, but just, thinking about or just tuning the genetics of oat. So, but if you see genetics of the oat, it is a complex genome and is a complex genetics, and also there are very not sufficient, genetic and genomic resources available as in other cereals in barley or wheat. So, so basically, if until 2018, actually, so we have only less than 1% genome was sequenced. But thanks to actually PepsiCo, they have just published the whole genome sequence in 2020, and which is a great resource now and helping oat researchers to do some other things. So, but also there are scattered efforts all over the world where there's genome sequencing is being done. But now with the help of the Pan Genome Oat project, and I'm honored to be part of that as well. So where, we will have probably within a year or so, the whole, sequence, pan oat genome sequences. So then we will have more resources to work in oat. So in my lab, actually, we are using many different strategies to explore oat genes and their function. So there are general strategies used in other crops as well. For example, first strategy, we are using a transposon mediated mutagenesis. We are very successful in barley originally, and as we have resources, we thought we should try in oat as well. And I will show you some of the results in that. And secondly, because the transposon mediated mutagenesis or tagging is sort of more random approach. So, and this is non-specific. So for targeted approach, so we also use RNAi. So, and now because of the recent popularity of the CRISPR, so we are also using gene editing for some specific targets. So in case of transposon mediated mutagenesis, as I just said, that we were very successful in barley, and using this system, we have genetically dissect many genes in barley, especially malting related genes. And so and as you know that this particular technology is based on the elegant research done by Barbara McClintock in 6 decades ago, and it's still relevant. So people have tried this system in many plant species, and where they successfully tagged many genes. And as we were successful in barley and we have the resources, we thought, why can't we also try in the oat? So how this system works actually, as based on the name AC and DS. AC is activated, DS is a dissociation element. So when so DS actually this element dissociate from a one genome position and then land to another genome position with the help of actually transposase activator. And so especially with the complex genome such as oat, it may be very useful, as this particular system has bias towards genes. So what we did is actually, we are now introducing all these AC and DS elements in oat. So basically how, in a system, how it works is, for example, this is just an example that if you consider this as a cell, and this pink color is a sort of a nucleus, and all those small threads are chromosomes. If we introduce or transform a plant or cell with AC and DS constructs, so you will be in the situation that DS and AC are together, and with the help of transposes, if it is expressing, actually those DS elements dissociate from its own place, and they can go either land to very closely linked position on the same chromosome, or can go to the different chromosomes. So basically these two attributes are very important in a way. Originally we want to develop randomly scatter this whole DS transposon in the whole genome, and in the end we can also integrate with that all the mapping information which already available. For example, where this crown rust gene is actually mapped, or other disease, rust disease, or other disease resistant gene is mapped. If our transposon is actually mapped close by, we can reactivate them again, just to target the closely linked genes as well. And we have successfully done this in barley, and we have actually dissected some QTLs which are related to the malting quality. So using this approach, we have to first transform the oat genome. So these are some of the constructs, AC and DS constructs, and some of those antibiotic resistance or herbicide resistance genes constructs there, which we are using actually. So this is a normal process actually of oat transformation. I don't think I need to explain that much. So, but we are basically using a park variety. So we use this mature seeds of park, and then from that mature seeds we develop callus, and then using osmotic media or different strategy, which is actually published by Peggy Lomo from UC Berkeley with the green callus system actually. So where there's no problem in actually regeneration. So basically using this technology, we can, develop all the transgenic, and in the end, we can analyze them. And so now I'm sure you know how the transformation is done. I'm not sure how many of you have seen a bombardment gun. And one of my grad student, Anis Fatmawati, she completed just recently her degree here from McGill, and she just prepared a small, this video. So. So basically what she's doing is that using this bombardment gun, and she's just, spreading this DNA of those constructs on those small screens, and those screens are actually attached to the gun, and which is actually run through helium gas, and you can bombard those DNA into the cell, and it's working beautifully in our lab. So, but before you start any transposon system in any species, generally what we do is we just test its efficacy actually in a transient assay. So this particular work is actually started by one of my grad student, Mahannad, and now actually being finalized by my postdoc, Dr. Joe. So basically, when we started to see its efficacy, we need to have two constructs here. For example, one construct is where the Gus gene is interrupted by DS, which is actually co-bombarded with the transposase genes. What happened with the transposase, the DS is actually moved, and then Gus gene is expressing. And if the Gus gene is expressing, and you can see those blue foci there. So we checked it, and it shows that it is feasible in oat, and we have done successfully this. And after that, we thought, well, why can't we then we have to make stable lines through transformation. And we use the exact same methodology which I just mentioned earlier, using these constructs. And in the end, we have developed all the stable lines which we can either cross, or in cases where AC and DS constructs are all together, we can then advance them further and then see if they are moving. So in case of transformation, I can't say that we can do 60% as Jorge just mentioned, but we have a transformation, close to 10%, in some cases we have seen 12 or 50% as well. But with this, with Biolifast selection system, we found approximating 9.5%. So originally we also were using the hygromycin system, but we didn't get much success there. Originally we thought that, hygromycin is not working, but lately we changed the source of the hygromycin. It started working with hygromycin as well, and basically transformation frequency is pretty much similar, between 7 to 12%. So once we have T2 lines or these transposon insertion lines, we have to check them at a molecular level. So what I'm showing here, which is a very important slide in a way, that there are 3 gels which you see. In the first gel, all the samples are in the same order. So first, in the first gel, we are showing that whether this is transposase gene still there or not. In some cases not there, some cases there, because we want to segregate away transposase gene, otherwise it will continue moving. In the second gel, we want to see if the DS is there or not, right? If somehow DS is there and transposase is not there, it means that line is a stable line. In the third one, so for example, if somehow the DS is moved to new position, right? And with these primers, there will be no actually amplification. So it means it is in the new position, it's not in the original place. So and basically that's what we have done. And you can see these 2 lines. This is just an example I'm showing to you, that these 2 lines are unique lines where the DS transposon is moved to a new different location. We also confirmed confirmed this further, by, checking if in the, empty donor sites doing a PCR, if there are small bands are there, and we found it and we sequenced it, and the results were confirmed, which indicates our ACDS system is working beautifully. So once we have those unique lines, we also do some other analysis to see in which genes they are present. But if I just want to summarize this part, we have seen approximately 5 to 17% transposition frequency. And why we were using this transposon system, because originally we thought the transformation frequency will be low, and in this way we don't need to do many transformations, and we can still do functional genomics nicely, and we can target many genes. So somehow the transposition frequency is pretty high, which is very much comparable to the Barley, which what we have seen. And as I said, that we also analyzed the flanking sequences of those DS element, and we found that they are in the unique positions, some of the lines I am showing here. So finally, this is indicating that transposon system is working great. So because this is we use the hexaploid, oat, and but we thought that in the diploid oat, it may be even working better because of the less redundancy, or it will be more useful for functional genomics. So then we start thinking, why can't we standardize the tissue culture and transformation system in Avena stygosa? And the seeds were kindly given to us by Dr. Rick Jalen and Dr. Jeff Mann from BYU. So we tested stygosa. The idea here was for the Avena stygosa is this is an 8 genome oat, because generally the 8 genome oats are higher in beta-glucan. So we checked it, and the system is working beautifully. So we standardized the transformation tissue culture system, and also the transformation system, and where we also found approximately up to 7 to 8% frequency of transformation. So now I will turn to the second strategy which we are using in my lab, which is more targeted way, for example, RNAi. So my grad student Anis Fatmawati actually started working on it. The reason is that we were more interested in the beta-glucan biosynthesis. We know there are cellular synthesis like genes are involved in beta-glucan synthesis, which are tested in Arabidopsis, which are tested in wheat. But very recently, so in my lab, we have seen there is an interaction between thaumatin-like proteins and beta-glucan. Then we thought we have an opportunity, because our transformation system is working. Why can't we check it if the thaumatin-like protein is also actually doing something in oat? So Anis actually started developing constructs and just finding first the homologue in oat, barley TLP8 homologue in oat, with the help of Dr. Nick Tinker and Tim Langdon. So and she developed those constructs, RNAi constructs, and then used the exact same way what I have just shown to you, the transformation. And she successfully developed different events, RNAi events, and then we also see again approximately 7 to 8% transformation efficiency here as well. And so this is I'm just showing one data from one of the line, and she checked in one of the line, she saw that there is a down regulation of the TLP8. So we are now currently actually doing other biochemical analysis on some of these different lines, and so some of them are now homozygous, and so we want to test whether it has any effect on beta-glucan or not. So we are also, as the gene editing is an important now part of any functional genomics lab. So I hired Thomas Donoso, who's actually leading in this gene editing effort, and we still, doing on the same gene, the TLP8, which I have just shown to you, with where in barley we have seen if the expression of the TLP8 is higher, the beta-glucan is lower, and vice versa. So Thomas started developing some of the gene editing constructs, and he successfully developed constructs which are using Golden Gate technology, and the constructs specifically which can target A genome TLP and C genome TLP and D genome TLP. And he did number of transformations and now using exactly the same method, but we also in this time we also added a GFP construct. So basically, at the callus level we have seen that transformation is working, and currently we are actually checking the gene editing efficiency, and so we extracted the DNA, so we will be using next generation sequencing to see deep sequencing to see if there's edits are there. So basically we are very, very encouraged what we are seeing at the moment. And so these are some of the strategies we are currently using for the oat improvement, and I'm sure we will be successful in developing more understanding about the oat genome soon. So with that, if I would like to conclude my talk here, so that in transformation we have seen approximately 7 to 10% frequency in transformation, and we have successfully transformed diploid oat strigosa as well, where we have seen the approximately similar type of frequency. And in the DS Transposon system, we have successfully developed that system as well, and we have seen approximately up to 17% transposition frequency, which will be very, very useful to develop to target many or tag many genes in oat. So TLP8 orthologs were identified in oat, and we have some other nice results in the different homologs where we saw that there's a possibility that the TLP8 probably be involved in the beta-glucan regulation. So we have successfully transformed the different constructs, including RNAi1 or gene editing one, and so currently analyzing the material further. So with that, I would like to thank my team, my lab members, but especially thanks to my collaborators, Nick Tinker, Vibui, and Tim Langdon. So Jeff and Rick, they have sent me this strigosa seeds and other senior members in the lab. And of course, you cannot do any work without the funding support. And I greatly appreciate the funding support from Prairie Oat Growers Association, POGA. They have helped us a lot. That's how we were able to develop such systems here, and also the gene editing for food security and environmental sustainability in new collaborative multi-institutional grant, which we have secured just last year. With that, thank you very much, and I am happy to answer your questions.
Thank you, Jaswinder. That was really great. Wow, there's so much information in these presentations. There's a lot of really fascinating work. We do have one question here from Dan Windrell. One of the perceived consumer benefits of oats has been their non-GMO status. Both presentations seem to indicate that we are moving away from that. Am I understanding correctly?
Actually, not really. And in a way, I can, first of all, when you understand the genome, you use the transgenic approach. That's what we are doing, so we know which gene is actually involved in what. For example, even in the beta-glucan synthesis gene, which I just said that maybe if the TLP8 is actually associated with beta-glucan, then we can make specific markers based on that TLP8, and use a breeding program. And we are doing the same thing for barley. And we have seen recently the TLP8 barley gene, which is already there. We found some interesting markers there, which may, which will be associated with, with this particular trait. That's one thing. And second thing, with the CRISPR technology, even very recently with the Health Canada, and USDA, if you are just tweaking the gene which is, we are not putting anything new. This is just for example, if we are tweaking 1 or 2 nucleotides, and if it changes something, right? So basically, Health Canada and USDA say they are not considered as transgenic as well. So, basically, but just to understand something, we need to use these kind of technology. Once you understand something, you can, employ or deploy those things in a breeding program in different ways.
May I add to this debate? First, in USDA, there's a limitation because currently they only allow you to edit 1 gene. Mm-hmm. So for oats and wheat, that's a killer. We cannot use it because we cannot use CRISPR in the low regulations because you usually need to edit multiple genomes unless you do a dominant mutation, but that is still limited. So even though CRISPR is less transgenic than other approaches, for some countries there is, and for exporting countries like the US and Canada, that is another concern. So in wheat, what we have done is we move away from the transposon tagging to use EMS mutagenesis, the normal EMS mutagenesis, but then we use exon capture and sequencing. So we had done that with the wheat tetraploid and hexaploid populations. We have generated a collection of 15 million mutations in wheat. They're in a database. People go there, you get 40 mutations per KB. So a gene that is 1 KB, you have 40 alleles. Usually you get 2 or 3 complete knockouts. And because that is CMS, is non-transgenic at all. You can put it in the field from the first generation without any permit. And we have been using that in our breeding program. So we already have several genes for resistant starch and other things already in the field as release varieties using that method. My advice to the oat community is get it done. It's not that expensive for a community. It cost us, when we did sequencing was more expensive, but right now we resequence not only the exon capture, we did a promoter capture in wheat, and we generated another 5 million mutations in tetraploid wheat in the promoters, and it cost us $200,000. So if the community gets together, and then it's just people go to the website, do a blast search, request the mutants that they want in promoters, in exon cap, in regions. We distribute the seeds. You can put it in a public place for distribution. Because it is an hexaploid plant, you can load it with mutations to a level that you cannot do in a diploid. In a diploid, you need to have 12,000, 15,000 lines to be efficient with tilling. But in wheat, that is exactly the same as oat. We have done 1,000 plants and get 10 million mutations in 1,000 plants. So that's enough. And exon capture, well, that's a couple of $100 per line. So doing 1,000 plants is not that expensive. Trying to do that by transposon tag, it will be impossible in wheat.
So basically, thank you, Jorge, for saying those things. But I also want to mention in the Canada, we have a different system. So even if you do something with the mutation, with the even chemical mutagenesis, they are come under PNTs, which is plant with novel traits. So we have to still go through the same process. But at the same time, I can, in the tilling one, yes, it has a lot of advantages, but at the same time, sometimes, we have to remove a lot of backgrounds as well. So if we are understanding or genetically dissecting something, so they are single copy or they are single transposons there. Both strategies have pluses and minuses, but I'm glad you mentioned about this USDA system. I was not aware that they have just for only 1 gene. So
yeah, hopefully they will evolve. And with the tilling, yes, we usually do 6-back crosses after when we used to use commercially our tilling mutants, but with 6-back crosses, we are okay.
Right.
Yeah, this is quite an interesting and touchy area for a lot of people, of course, and a lot of it is people's perceptions.
Yeah, I'm a breeder. I have my own breeding program, so I realized that if I want to get something into the field with transgenics in wheat, will not happen in the next 10 years.
Yeah.
So I don't have that time. I'm too old. So that's why we use the EMS mutagenesis.
Actually, I am little bit more hopeful. I think I can consider 5 years, but because of the gene CRISPR stuff. But I understand what you are saying. So basically, but what I am trying to show, how to understand the things. This is just a, it's not, I'm just not trying just to develop immediate a variety, but understand something and then use to develop a variety.
Yeah, the point is, once you create a resource, right now, if you have those lines, you still need to have a distribution system and people need to have a weight. So even if you create the transposon tag things, you will need to sequence all, as they have done in Rhizome or Arabidopsis, you still need to sequence the insertion points of all the transgenic lines to get it done. So it's, once you create a resource that is a web-based, that was our limitation. We first did tilling and then had to distribute the DNA and people were not using it. But now that it's just, you go, you do a blast, you get your list of mutation, request the seeds, you get the seeds. Now it's a system that the community can use very easily. And people without any resources can access mutations in any gene of wheat. So I think it really democratizes a lot the use of functional genomics in a crop, if a community makes the investment in those type of technologies.
Sure. Thank you. But with Phil Bergedzer, although he is not no more here, but we developed something similar system in barley where people can order some of those lines and do the research.
No, I think it's important for things to be open to everyone, and the transparency would certainly go a long way to helping the image of some of these technologies and so on. So are there any other questions? I see that Grant has put a link in the chat for the next Speaking of Oats webinar. Everyone can take a look at that. We are close to time. Would anyone else like to ask a question or make a comment? Oh, Steve Eichten says, cool stuff. Yes, it very much is. It's a very complex area. It's also very fascinating. So thanks for that, Steve. Okay, so I think, Jaswinder, if you can stop sharing your screen, I can put up the information for next month. This one. And then Tom, if you're still there, perhaps you can take us out.
Yes, that sounds good. Yeah, great presentations and great number of attendees on as well participating. So good to see that. So next month, coming up in May, another event, hoping to get everybody to sign up again and participate. Improving heat stress tolerance in Minnesota, adapted oats using ecophysiological approaches. So looking forward to that. I've heard some of this from Kevin Smith over the last 12 months. So looking forward to hearing more about that. So I think we're gonna be done here, wrapping up.
And thank you very much to the presenters today. Really excellent talks.
No, thank you for the opportunity.
Yeah, very good.
Thank you.
I do actually have a question here from Ben. How many people attended the meeting? And I think in the middle, I think we had 60 people.
I was going to say
great.
about 60. Yeah,
That's excellent.
Yeah, so that's great.